Hello everyone, welcome to today's Focus On session provided by GenQA. Can I please ask you to mute your microphone if you are dialing in live? We've had a number of questions in advance of. If you do have a burning question or require clarification on the presentation, please can you use the chat box on the website to submit a query, and this could also be useful if you're experiencing any technical difficulties. We've had an unprecedented response to this webinar with nearly 800 registrants. So unfortunately, we might not be able to respond to everybody's question, obviously due to the time constraints. So today's presentation is a focus on the International System for Human Cytogenomic Nomenclature and the new 2020 edition of the book. I would like to introduce Dr. Ros Hastings, who has many years experience with ISCN, and she is an author of the 2020 edition of the book. Ros is a part-time consultant with GenQA, and she will be giving the first part of today's presentation. So I'll pass you over to her now. Thank you.
Thank you, Melody. Good afternoon, everybody, or maybe morning in some countries. What we are going to focus on today, if I can get my slides to work, there we are, is some of the common misconceptions and frequent errors that we see through EQA in GenQA. A summary of the changes for the new version of ISCN 2020, obviously not in a lot of detail because there are a lot of changes for 2020. A new EQA we're going to be offering for 2021. We're also going to give you a little quiz, which is interactive, which I hope you'll stay on to participate. And then there'll be a question and answer session. But let's talk about some of the common misconceptions that people have about ISCN. The first one is there's no need for you to read the whole book. I'm afraid you do have to read the whole book. We have tried to make it a little bit easier by summarizing some of the information at the beginning of the book, but if you really are going to benefit from it, then you do really need to read the whole book. There is no good just looking at the section with the type of abnormality that you have found, cytogenetically or molecularly either, because it's a book whereby you build the information. So you do need to understand every aspect of different structural rearrangements and how you report them. Some people said that the ISCN isn't logical. I'm going to dispute that and say it's very logical, because if you do read the book in its entirety, then you will see that there is a lot of logic in the way that everything is presented, and I will try and go through some of that in a short while. Another misconception is that it's helpful to the reader of the report to describe polymorphisms in ISCN. As these are not abnormalities and have no clinical phenotypic effect, the new ISCN 2020 clearly states that you do not describe polymorphisms. They may be used for other purposes in hematology, for picking up chimerism, or it might be used in some constitutional ways, but it is not there to be described to the clinician because it's of no clinical relevance. And again, another misconception is that normal FISH results cannot be reported in one string. And again, there are examples in the book whereby if the same number of interphase cells were analyzed for the different probe hybridizations, you can report them in one string. So I'm going to hopefully now go through some of the basic rules in ISCN and distinguish between myth and fact. The basic rules. The new one for ISCN 2020 is that every abnormality, regardless of technique, is now described from Peter to Cuta. So you write down what you actually see. Cell numbers are given for mosaics in constitutional results and clones in hematological results. The chromosome number order goes from the lowest, i.e. chromosome X, Y, then 1 to the highest chromosome 22. Numerical, then structural aberrations are given in chromosomal order, and structural aberrations are listed alphabetically, first of all in chromosome order and then alphabetically. So a del comes before a duplication, for example, and comes before a translocation if it's in the same chromosome. Obviously, if it was in chromosome 1 where you've got a translocation, that would come first before a deletion that was perhaps in chromosome 22. In fish, the numerator is the number of abnormal cells. That's the top figure, the 12 in this example. And the denominator is the total number of cells, which is 50. For fish, only for abnormal cell lines, you list them individually for the same probe. And contigs must be given in the full ISCN or text. You've got an option, you can do either or, but you do have to explain what the probe is composed of. Normal controls are not given in the ISCN except when it's sex determination or if the actual control probe is abnormal. You can combine short and the detailed form of the ISCN if you've got a complex rearrangement. So if they've got multiple abnormalities and one is more complicated, you can put that in the detailed form. When multiple techniques and mosaicism is found, you put the G-banded result first, followed by ISH, followed by NUC-ISH, followed by array, followed by RSA, and then sequencing. Obviously, if you're not doing all those techniques, you don't have to put them all in, but that's the order they actually all go in. So you list all the G-banded results, even if it's a mosaic, before you go on to the fish, before you go on to NUC-ISH, etc. You can now, and you could in 2016 version, list the results of the different techniques on a separate lines. And you can tabulate complex results, but you do not tabulate simple results. And for arrays, you can describe the mosaic either as a percentage, like 60% is 0.6 or 2 to 3. You can use either option, but you do need to describe if a mosaic is found. The RSA ISCN is not dependent on platform. So depending on the type of technique used will depend on whether or not you either give nucleotide positions or you give a kit name. And again, for mosaics, you can write either 2 to 3, and if you can quantify the mosaicism with the technique that you are using, you can put it as 0.6 if you wish. So there are a few more options, and hopefully we've made this much more clear to participants so that they can use the ISCN effectively. So what are the common errors that we see in GenQA when we're doing the external quality assessment, EQA? Well, for normals, we often find that people mix the interphase with the metaphase fish. And if you've got a normal fish result, you can use ×2 for both the interphase and the metaphase ISCN. The metaphase fish includes the probe location, and it's optional whether you use it for interface fish. There are examples in the 2020 where they are given and other examples where it's not. For sex determination, we've now specified that both probes must be given, i.e. you give X and Y, because you need to know whether both are absent or present or what combination you have got there. For abnormals, it's to remember that for metaphases, you use the plus sign or minus if it's missing. For structurally abnormal metaphase fish, you only describe the derivative chromosome or the abnormal chromosome. And if it's mosaic and they're both abnormal, you have to describe both cell lines. In interphase fish, you use ×3 ×1. You don't use the plus and the minus, and that's where often we're finding people are making mistakes. And for break part and fusion probes, you describe the signal pattern first. I'll just quickly explain what I mean by the signal pattern. So you actually, for both break apart and fusion probes, you are describing what number of red and green signals you're seeing before you describe what the rearrangement is. Other common errors in the fish probes are to do with the fish probe order. The probes are listed in chromosome order from Peter to Cuta. And for the abnormals, again, they are listed in chromosome order. And the big change for everybody is that sex chromosomes, just as they are in a karyotype, are listed first, both in fish, in arrays, and sequencing. Interphase, for the fusion probes, you're following the chromosome order of the probe sets. So because the probe sets are obviously multiple chromosomes, it's the lower chromosome you're looking at first. So for BCR able, you're looking at the able because that's the lower chromosome number, and that is given before, for instance, ETV6 RUNX1. So some of the common errors are to do with actually how do you define a clone? And the same might be also the same for cell lines. So a clone in hematological terms is 2 or more cells with the same gain or aberration. The exception to this is when a single cell can be reported is when the same clone was found at a previous or a follow-up study of the same patient. And likewise, for a loss, you have to have 3 or more cells with the same loss. And the exception again that's in ISCN is you can have 2 cells with identical losses if one or more of the chromosomes, when there's also a structural or a gain in the analysis that is considered to be clonal. So for instance, if you've got loss of, say, chromosome 22 and you've got a 9, no, let's not use that example, and you've got an inversion 16, then if you've had loss of 22 and both cells have an inversion 16, you can report it. The other problem sometimes happens is about what order do you write the clones or the cell lines, and it's slightly different between constitutional and hematological. In constitutional, cell numbers are only given if the result is mosaic. And the largest cell line is always listed first. Normal cells are always listed last. So you don't conclude them as the largest cell line. In hematology, cell lines are always given. There's no exception to that. And normal cells, like in constitutional, are listed last. The stem line is always listed first, regardless of size, and the side lines are then listed. When there are 2 stem lines and multiple side lines, the use of SL1, SL2, SDL1 and 2 can reduce the clarity, and therefore we have recommended the use of IDEM for such cases when you've got a complex karyotype. It's very difficult with the use of the internet and lots of publications and people coming working in different environments that we don't always use the same terminology, and sometimes the terminology changes just because of common usage. However, there is some terminology that you can't change because it's defined by genetic mechanisms. And so it's really important that you use the term recombinant when there is a recombination event that has occurred, and you use DO when it's a derivative chromosome as a product of an original arrangement, whether it's de novo or segregated from a parental rearrangement. A recombinant is a structurally rearranged chromosome with a new segmental composition resulting from meiotic crossing over. And so that is a particular genetic term. Derivative chromosomes are products of an original rearrangement and they segregate without further change. And so the two should not be mixed up. Add can also be used in ISCN, but this is used when additional material is of unknown origin. But as we all know, when we've seen it being used, it can be misleading because there can be loss of material even if you use the term add. So DER is really a better term to use, although we have not stopped people from using add. So what about breakpoints? One of the things that you're allowed to do in ISCN is if another technique delineates the breakpoints further, you can change the breakpoints. So when Fish or Array extend the breakpoints from G banding, it is optional whether or not you change it in the G banded karyotype, but you do have to put the new breakpoints in the Fish and the Array part of the ISCN. It's also optional whether or not you repeat the breakpoints in the Fish ISCN if it's exactly the same as the G-banded ISCN. So you've got the two options. In the first option, we didn't repeat the breakpoints, and in the second, you have. Obviously, for clarity, it is much easier to see the first example. One of the things that some centres have difficulty with is actually working out how to do composite karyotypes. A composite karyotype is a clonal karyotype. It's just you sometimes may also have random losses and you ignore them. So it's not the same as a random loss. So if you had these 5-cell lines, the composite karyotype would be 43 to 46, XX -7 because you've got 4 cells with a -7, and +8 because you've got 3 cells with +8, and you can say it's a composite. The first one is not included because they're random losses and there's no clonal abnormalities there. The second, third, fourth, and fifth all have part of this composite, and because you can put them together, you can know that there are 4 cells there that have -7, so that fits the clonal definition, as does the additional chromosome 8. There is a big change now for the sequencing ISCN that Johan has been very heavily involved in, who helps with the HGVS, that's Johan Dunham. In the sequencing ISCN, there are 2 lines. The first one is to describe the rearrangement from a cytogenetic point of view, and the second one is to give the sequence ISCN. You only use both lines when you know the type of rearrangement. If you don't know the type of rearrangement, you just give the sequencing. However, one of the changes for 2020 is you now have to use the reference sequence version number. So if you have a rearrangement that could be an inversion or a marker or a result of an unbalanced insertion or imbalanced inversion, then you don't know what the actual sequence is from a cytogenetic point of view, and you can only give the HGVS part of the ISCN. So let's look at ISCN 2020. ISCN 2020 came about through the Standing Committee, and these are individuals from the different continents, 2 from each continent usually, who go through all the submissions of the changes that are required and review the whole book. It's published by Karga, and in 2020 we had a major revision. The big change for this year is there's now an online version available. So when you get the book, and there's a picture there of the book, you will get on the inside cover a unique code that will enable you to log in so you can access the online version for 12 months. And GenQA will expect participants to use the ISCN 2020 from April 2021 onwards. So as I mentioned, it was a major review. There was a few corrections that had to be also incorporated from that were in ISCN 2016. We had 222 suggestions, which we went through over 4 days. Things that we've changed, very difficult to put them all in one presentation. You will note when you get the book that as in 2016, the grey lines to the side indicate some of the changes. It doesn't indicate where there is new examples, but it does indicate where there are changes in the ISCN text. We've added general principles table for the different techniques on page 37. In hematology, one of the difficulties is for laboratories on how you count rearrangements, and that is given on page 97, and that was through input from GenQA from the assessors who mark the submissions, and they've basically looked at the abnormality type and given examples and stated how they think how many chromosome abnormalities you should count it at, whether it's counted as 1, 2 or 0. The array chapter in chapter 14 has got more examples, and we now got polar body ISCN included. The RSA chapter was incomplete really in 2016, and we've given you more examples. The sequence chapter has been revamped, and again there are more examples. We've removed the CGH chapter, and the UPD section that was in 2016 is now put under array. There's further clarification of the text, and hopefully we've got more consistency across the board. So just some of the things that have, where we did have inconsistency, where you put the sex chromosomes in the ISCN was different between karyotyping and the molecular techniques. Whether you wrote from Peter to Cuta also varied, the cell numbers, mosaicism, and genome build. So we've tried to make this much more consistent. The basic rules we've now sort of summarized at the very beginning of the book on page 37. You will notice when you get the book there are 2 page numbers. This was not what the authors wanted. It was insisted on by the publishers because it's obviously reprinted from another journal, and hopefully going forward this won't happen again. So the basic rules, general principles for the different techniques are given on page 37, and it looks something like that. Sex chromosomes will be reported first for all techniques. We describe the abnormalities from Peter to Cuta for all techniques, so you're consistent with the array and sequence nomenclature. Nucleotides can be separated by commas in the short form. It is optional. If you don't wish to use commas, you don't have to. When an unbalanced derivative is inherited from a balanced rearrangement in the parent, DMAT, meaning derived, and DPAT will be used. As if you just use mat or pat, it could imply that the parent is unbalanced. In fact, you don't know if they're balanced or unbalanced, which is why we made the distinction. It does state that heterochromatic variants are not given in ISCN. There are more P-arm examples, and we've included in the FISH chapter some tricolour probes because that didn't exist before, and we specified which HGVS version has been used for sequence nomenclature. This is the another page which shows you how to count the chromosome aberrations. I'm afraid it's very pale. I've just taken it as a photograph from the book. There will be some more changes to ISCN 2020. Unfortunately, we've already found one error, which happens on page 69, and the long form of the ISCN is incorrect. We've put P34 instead of P31, and we say it's to Peter when it should be to Cuter. Also, since publication of since this document went for publication, we've put in the text that the fusion gene is BCR-ABL1 for any of the fusion genes, which has been standard practice. However, the Hugo Gene Nomenclature Committee has stated that fusion genes need to be written now as BCR/ABL1, as the hyphen is used for read-through transcripts. If you want to find the paper, it's in Bruford et al. In Nature Genetics, and they have acknowledged this is a change from what has been common practice. So I'm afraid, even though we've got a brand new ISCN, there's two new things you have to take into consideration. We're currently in discussion with the Karga, who've got the website with the ISCN, whether we can, how we're going to put new changes and on the ISCN 20, so it is kept up to date, even if the books obviously have to stay as they are. I'm now going to pass over to Melody, who's going to talk about the ISCN Pilot EQA.
Thank you, Roz. So in 2021, GenQA is pleased to be able to offer a new Pilot EQA, and this is going to be based on ISCN. Thanks. So this is a new exploratory pilot, and we are aiming this pilot at laboratories who report results using the ISCN, and also clinicians who receive reports containing cytogenomic results to help people understand the use of the ISCN. At the moment, we are aiming to offer a minimum of 10 cases, and this EQA will be purely online. It's not to test the analysis or the interpretation of the abnormalities. It's purely to assess the use and application of the ISCN. So we will provide karyotype ideograms or fish images, and we will point out the abnormalities, the breakpoints, and the fish signal patterns. All this information will be provided for you, and all you will have to do is simply provide the correct ISCN. There's been a good response to this Pilot EQA, and we've had some initial feedback already. And there's people have asked if we can also provide microarray and QF-PCR questions. So that's feedback that we are going to take forward and include in the 2021 EQA. So this EQA is designed to test your knowledge and application of the ISCN 2020. So the 2021 EQA will open in October. It will all be online, like I said. You will be able to submit your ISCN answers using a proforma, which we will provide on our website. You will have 5 weeks to submit your results, and the results will be assessed by a panel which will include ISCN experts. There will be no performance monitoring or appeals process at the moment, because this is a Pilot EQA. And you can enroll for this EQA from now onwards using our website, which is listed there, www.genca.org. So those of you who are not familiar with GenQA, this is what our front page of our website looks like. And you can see at the bottom links to join GenQA, and there's all the options to find out more information on our EQAs and find out more about us. Once you have become a member of GenQA, you can enroll in the various EQAs, and this is the screen that you'll need to click on to enroll in the EQAs. If you have any trouble with registration or enrollment, please contact us via our email address, and we'll be able to help you with any questions. So the Pilot EQA will include, as I said, a range of techniques, karyotype, fish images, microarray data where we can, and it will also cover a range of constitutional and oncological cases. It's online, it's aimed at laboratories at this point, but because it's online, it can be accessed by multiple staff members. So if there are some departments who are specialising in constitutional, those staff members can answer the constitutional questions or the fish questions. Everybody can participate from your laboratory for this laboratory-based EQA. We recognise that some laboratories are specialists and will only be able to provide services for just the constitutional cases or the oncological cases. And so we accept that you might be able to just want to participate in a subset of the questions, and you'll be able to explain why you've submitted a partial submission to the assessors in the additional comments. So that's the laboratory-based EQA, which we will open in October 2021. We're also very excited because we are working with our software developers to develop bespoke software for individual competency assessment, in addition to this laboratory-based EQA. And this is what we've been referring to as GTACT, which stands for Genomics Training Assessment and Competency Tool. This will allow the ISCN to have personalised participation according to a staff member's knowledge and experience. So we hope to provide various different modules, including different technique types and sample types, and also different levels of complexity. This should hopefully be useful for training new staff members, as well as assessing the individual competence of the existing staff members. This will be provided online, it will be available at any time, and the results will be given as instant feedback. GenQA already offer some GTACT modules, and this is the website page where you can access those that already are offered. For example, we have ongoing modules in sample reception and report authorization. So now to the fun bit, if everybody's still there. This is the first time that we are offering an interactive part of our webinars in GenQA. So please bear with us if we have any technical difficulties. Having explained the various rules of ISCN, we thought it would be interesting to just pose a few different scenarios and questions to you, and you can vote on what you think would be the right answer. So the questions will pop up in a poll format, and you'll be able to submit your response by clicking on the choice of answer which you think is correct. I'd like to just reassure you that responses will be anonymous. Once the poll has closed, we should be able to see just the percentage of participants who voted on the various options. So if we go to the first question, Ros, I shall read it out. So what we want to know here is which of these options is correct. So we've got karyotype results here. There is one normal chromosome 21, and there's a rearranged chromosome where there are 2 copies of the long arm of chromosome 21. So how would we write this karyotype? So if I click on here, you should be able to see the question. And if you click on the round circle for the option that you think is correct, I'll give you a short time to think about it, and then I shall close the poll. So remember, there's one normal chromosome and one chromosome with 2 copies of the long arm of chromosome 21. And here we can see the results. So 41% of our participants have voted for the first option using the isochromosome, and 59% have gone for the second option. So if we go back to the presentation, Ros can explain the rules and why the second option is the correct option.
Right, and the reason is it's to do with mechanisms, and that is the isochromosomes. You don't have an additional chromosome 21. It's the original one chromosome that's basically splayed its arms and become through a misdivision around the centromere. So if you use you have to use DIR, because unless you've proven it is an isochromosome, this is the correct nomenclature to use.
Thank you. Okay, so we've got a few more questions. So if we move on to question 2. Can you click on the presentation? That's it. So we've got some fish nomenclature here. So we've got a karyotype where there are 2 cell lines. One cell line is showing monosomy X, and one has an abnormal chromosome Y. And the fish has been used to try and characterize that chromosome Y. So which option is correct? You should be able to vote now. So essentially, we're looking at whether you need to include the fish that was seen on the monosomy X cell line, as well as the fish that you could see to characterize the chromosome Y. Okay. So we've got 63% of participants have got the correct answer, which is using the fish to describe both cell lines. So I'll just hide that. There we go.
Right, to explain, the problem with the first, the incorrect answer is twofold. You've got 2 cell lines here. If you just give the fish for the Y, then the 15 cells that you're not talking about actually could be normal, because you haven't specified that actually they have only got a single X chromosome. So you do have to describe both the cell lines that are present, and you have to include the X in the description of the cells that have the iso Y. So this is a situation where both cell lines are abnormal, so you have to describe them both in fish.
Thank you. So moving on to question 3. So here we've got a structural rearrangement of a chromosome 5 and a chromosome 9. There's been a translocation between the long arm of chromosome 5 and the short arm of chromosome 9. The translocated chromosome 5 also has a deletion of the Q arm from the breakpoint of the translocation. So how would you describe this karyotype? And let's close question 3. And again, most people who've voted have gone for the correct answer.
So again, you have to really describe what you're seeing, and it's important that you it's not a translocation with a deletion. They've got 2 abnormal chromosomes effectively, although there was a translocation. So you need to describe the derivative 5 and then the derivative 9.
Thank you. So the next question is with a fish probe that's a triple color probe. So this has now been included in the 2020 ISCN. This triple color probe is for the MECOM gene, and it's being used here to detect a rearrangement. We've got a split signal for the MECOM probe. We've done this fish just on interphases, so we've just got the nuke-ish nomenclature. And which option are we going to go for? I'll just open this poll. Unfortunately, the word limit on this question didn't allow the full answer to be put in the answer. So I've included the first option is in the question. So if you want to vote for that one, click the answer above, and the second option is just below. So do we describe the number of signals of the various probes first or not? And how do we describe the pattern of whether they're separated or not? So had a much more even split now. Is it the first option? I won't read it all out, obviously, or is it the second option? I can tell you that the first option is the correct one. And I'll just hide this and Roz will be able to explain.
This is probably a very unfair question to give you, because unless you've got ISCN 2020, you wouldn't have a clue how to write it. And so it's interesting that you have given it you're pretty split on it. So basically you will have 3 copies of Mecon. And so the second one is definitely wrong because you've only got 2 copies there, because you've got a split Mecon signal. So you will have 3 signals. And because it's a triple probe, you've got to refer to it in the sense that Golem and Mecon are together. They've Mecon separated, but Mecon and Mecon is together. So you just need to think about what's actually happening from a fish perspective and what you're actually seeing. So that's why we've described it this way. I've not explained it very well, I'll be perfectly honest with you, but I hope you can see when you look at the book that there are quite a few examples for triple probes and just how you do write them. And if you do have any queries, you can always get in touch with anybody on the ISCN committee.
Thank you. That was a tricky one. So question 5, again, now we've combined karyotyping and fish. We've got an additional abnormal chromosome here, which is a dicentric chromosome involving the P-arm and centromere of chromosome 15 down to Q1 1.1. And the fish has helped define these breakpoints because we can see that there is no SNRPN signal on this dicentric chromosome. But how do we describe those fish signals? Just like to say at this point, thank you for everybody voting on this, because it's really interesting to see the results and explain the thinking behind the answers. So let's close this one. So we can see a lot more confidence in the answer here that the second option is correct.
This option, this case is now actually in the ISCN. And the thing to remember is the first example just tells you what the signals are, but it doesn't tell you their position, which isn't very helpful, because in fact they could all be on one chromosome arm. I mean, it'd be unlikely, but they could be. So what you have to do is you have to describe what you're seeing from Peter to Cuta, and you have to remember that it'd be the reverse way up or reverse way round for the lower Q arm. So you're going from D15Z1 down to SNRPN, and then where it merges, you're going back from SNRPN back up to D15Z1.
Thank you. So now moving on to some more hematology-based questions. Question 6. Here we've got 2 clones. One clone has the 411 translocation, and that's found in 6 cells. And the second clone also has a derivative chromosome 4 from that translocation, and that was in 8 cells. But which way round do we write these clones? Okay, I'm about to close this one. That was interesting. I could see people voting, and initially everybody had gone for the right answer, and then as people were thinking about it more slowly, they evened up. So the majority have got the correct answer. It is the bottom option.
Right, so just to explain, the stem line goes first, not the largest clone, and that's why that's the way round that it goes. Now the next example will challenge you even more.
Okay, so this is a similar scenario. This time, this could have been the same patient. We're not seeing that stem line. So we've got 2 abnormal clones, and we've got an additional chromosome 19 in one of them. And we've got that clone that we had before with the 411 translocation and the extra derivative chromosome 4. Which way round do we write this? Okay, I think Rosie's going to be surprised at this one. Not as hard as she thought.
Oh, that's good.
81% have got the right answer.
Oh, well done, everybody. That's fantastic. So you've all basically remembered that D goes before T because it's alphabetical if there's no stem line clone. Stem cell clone, sorry. Well
Brilliant.
done.
Okay, we've got a couple more. We're moving on to some region-specific questions now. We've got quite a few options on this one, so we've worded this one slightly differently. We would like you to click on the incorrect answer or answers. You can click on more than one answer if you want. So which is incorrect? Okay. So we're looking for the incorrect answer, and the incorrect answer is the one that 49% of you have voted for. There was only one incorrect answer.
So this is just to show you how to put the X chromosomes and Y chromosomes in the new ISCN. And you are allowed to put RSA18. You can abbreviate it, although you must put in the text what other chromosomes you've actually tested for if you did actually do that with, say, QFPCR. The one that is incorrect is the one that says RSA18 times 3, because you have to do it in numerical order. So 13 goes before 18, and then 21 has to go after the 18.
Okay, and then the last question is looking at combining karyotype and FISH results. So we've got 2 different cell lines here, both abnormal. FISH has been done to further characterize the abnormal Y chromosome. Oh, sorry, we have got a normal cell line as well. So we've got 3 different cell lines. So which of these is correct? Okay, so let me just close this one. So we found this one a bit more tricky. We've got quite an even split. It is the top answer which is correct. So the majority have got the correct answer. And that's the last question.
So the issue here is that you always put the G-banded result before you put the FISH. The FISH goes after, regardless of whether there's 2, 3, 4 cell lines, and you're only giving the FISH for one particular aspect of the cell line. It doesn't go after the cell line you're talking about. It goes after the whole string of G-banded ISCN.
Thank you. That's the end of the quiz. Thank you ever so much for participating in that. And that brings us to the end of our presentation. Those are our contact details there, and now we will move on to, yep. The email address is obviously also on our website. We'll move on to a quick question and answer session. I'm aware that we've not got long for this. As I said before, we had quite a few questions submitted in advance of the webinar today. There's been a few questions on the chat, which I'm just going to have a quick look at. So Ros, put you on the spot here. Are you marking against ISCN 2020 this year?
Yes, we will from April 2021. We usually give you 6 months after the publication of any new changes, and this was actually published, I'm afraid, in November. So 6 months is up in April. If you've got any queries, you can contact me.
Thank you. We've had a couple of others which I think we've already covered in our presentation, hopefully. So highlighting the main changes in ISCN, and let's have a look. The correct ISCN for PCR aneuploidy tests, and we had a question on that, obviously, and how you would describe mosaicism. Ros covered that at the beginning. Whether you put 2 to 3 or 0.6, you can do that. How about my interest is on microarray ISCN for hematology and oncology results. We currently use a table format no matter how complex or simple the result is, as it's a format our clinicians are familiar with. Will this be officially made acceptable?
Right, well, on page 126 of ISCN, I think, I'm not sure if that's the new version or the old version. I wrote this down actually. You can give complex results in tabular form, but you have to give the chromosome band, whether it's loss, gain, amplification, or region of heterozygosity. You have to give the genome build, and you have to give the genome coordinates in that. So that you are allowed to do it, but it's only for complex ones. You can't do it for all results. You can't do it for simple results.
Okay, that's brilliant. You've actually answered the second part of the question there as well, which is what detail will be required. So, no, that's brilliant. So covered it all in the first bit. Another one is about the recently published guidelines for the. Sorry?
It is page 126.
Oh, yeah. The Hugo gene nomenclature guidelines. So I think we've covered that in the talk.
Yeah, that's a real frustration that they've made that decision, but I think we're going to have to all go with it, unfortunately.
It's just unfortunate that the publication dates are so close together.
Yes.
Okay, is it the band proximal to the breakpoint, so the band that is not translocated that's used in the notation, or is it the band in which the actual breakpoint is?
It's the actual band in which the breakpoint is.
Brilliant. And a fish one here, Ros. How do you describe a normal metaphase result using double centromeric probes like the 1422 or the 1321, which cross-hybridize? So a normal metaphase.
You would have to put both of the probes in. So you'd have to put, you can either do it just by putting open brackets D13Z1 / D21Z1 times 2 close brackets, or you can even put the breakpoints in. But if you put the breakpoints in, it becomes messy. Actually, hang on, is this interphase? It's metaphase.
Metaphase, yeah.
You have to put breakpoints in. So it's very messy. I'm not sure if these probes still exist, actually, do they?
I think they do, for the centromere probes, which
right. Okay, so
yeah.
you have to put the breakpoints in, which makes it what, 13P11.1 Q11.1 /21P11.1 Q11.1, and then open the brackets with the D13Z1 / D21Z1 times 2 close brackets. That is cumbersome. Sorry, that's it.
Probably easier to see if it's written down. Thank you, Ros.
Yeah, if the person if they're on this call, if they want to email me, I can reply.
Yeah, I'm afraid we've got a few more other questions that I think we're just going to run out of time for, but we will try and respond to these questions. Some of them I think we have covered in the talk, and some of them are a bit more specific to particular scenarios. But as a registered participant, you'll have priority access to this webinar for 1 month. So anybody who is unable to join us live will be able to review the webinar. You can look back at it again. If there's any members of staff who are unable to join us today, they'll be able to view it. We'll send a link to you because see, we welcome your feedback, particularly on this one, whether you enjoyed our interactive quiz aspect. That's the first time that we've done that, as I said. Those are the details to be able to view the webinar on our YouTube channel. If you've attended today, you'll be emailed participation certificate. And obviously, as I've said, if you need any further information, please see our website or contact us directly by email. And I'd just like to mention that the next Focus On topic, the webinar will be held in February, and our topic in February will be quality and using EQA to improve your diagnostic service. Hope you all enjoyed today's presentation and found it useful. Look forward to you enrolling in our ISCN EQA in 2021 and the feedback on that. And we'll keep you updated with our software development. So it's goodbye from me. Anything else to add, Ross?
No, just thank you all for joining. And if you would like to give us any feedback by emailing us, feel free to do so. We'd like to improve. And if there's more things we can offer and help you with, then that's what we're here to do. So thank you.
Thank you. Bye.
Bye.